If we talk about the nervous system and we move on to talk about muscular control, one of the things that we have to talk about are skeletal muscle receptors, part of the afferent division, sensory information. And there are muscle receptors that are aptly located in your skeletal muscle that send information to the brain about the degree of contractedness or stretch of individual muscles. Now, your brain can take this information, integrate it, and basically give you a sense of spatial awareness, right? So, in one of the videos I talked about, you could raise your hand, close your eyes, and take your other hand and give yourself a high five without having to look at your hand. Well, how is it that you know where your body is at in space? And there are two types of receptors that we'll talk about specifically, and one is known as a muscle spindle receptor, and the other is known as a Golgi tendon organ. Okay, so muscle spindle receptors and Golgi tendon organs. So the muscle spindle receptor, if we look at, is found deep within the muscle. So we look at a muscle, I'm going to draw, let's just draw a tendon here. Okay, and then let's draw a tendon down here. Okay, and if we draw the muscle, we have muscle fibers like so. Right, form that nice little belly. Okay, but we have muscles, sort of muscle fibers superficially, but then I'm going to draw in red some more muscle fibers internally. This is a view of what we call, these are intrafusal muscle fibers. And then in orange, right, these are our extrafusal muscle fibers. Okay, and what we have is that there is a spindle organ, I'm going to draw this in blue, that sort of like wraps around or spirals around the intrafusal muscles, right? And so when these muscle fibers are stretched, this is going to send a signal out saying, hey, that muscle is stretched. Okay, so this is going to send that signal out to the central nervous system. Now, coming down into the muscle itself are motor neurons. Right? And there are two different types of motor neurons that we talk about. There are alpha motor neurons, which are going to innervate those extrafusal muscle fibers, right? So your alpha neurons are going to sort of send signals to the extrafusal fibers. Then there's a secondary type, which we'll draw on sort of, let's do yellow. And they come down, right? And they innervate the intrafusal, right? And so these are what we call your gamma motor neurons, right? Your gamma motor neurons, okay? So it's my signal for gamma, right? So you have alpha motor neurons, you have gamma motor neurons. So when these intrafusal muscle fibers are stretched, they're going to send a signal to the central nervous system saying, hey, this muscle is being stretched. And one of the things that you don't want to happen, right, is you don't want it to be stretched too far because that will tear the muscle fibers. So there's a reflex that comes back that is going to say, hey, this muscle is stretched. We don't want it to be stretched. So let's stop the stretch or the lengthening of that muscle. So if we want to stop the lengthening of it, right, we have to contract it. So there's something that what we refer to as alpha gamma coactivation. So what that means is that when this sends a signal out to the CNS, signals are going to come back and it's going to activate the alpha motor neurons, but it's also going to activate the gamma motor neurons. Okay? Now, the reason that alpha gamma coactivation is important is because what would happen if I took these fibers out here, right, and I shortened these fibers out here without doing anything to these fibers here? right? So what happens if I shorten the extrafusal fibers, but I didn't shorten the intrafusal fibers? It would look something like this, where you would have this shortening of these fibers. Let me draw the shortening of the actual muscle. So something like this, where those extrafusal muscles were shortened. Right, so we had shortening of those extrafusal muscle fibers. But what would happen if we did not shorten the intrafusal fibers? Notice that they would go slack like this, right? And so if they were to go slack, right, there's no stretch on them. There's no tension on them. If there's no tension on them, this gets shut off, right? So we don't want that to happen. So what we do want to do is we want to activate both of these at the same time, right? But what we don't want to happen is we don't actually want these fibers here to shorten themselves. So what happens is that the shortening in your intrafusal muscle fibers, the shortening happens here at the polar ends, right? So all of the motor proteins are located at the polar ends of the fibers, and the shortening happens at the polar ends, right? But that keeps this muscle, these intrafusal muscle fibers, taut, so that this muscle spindle receptor can send a signal to the central nervous system. Now, the other thing that happens, right, is when this gets sent to the central nervous system, imagine that you have a bone and a bone, right? So this is your joint. And we have a muscle, right, that runs like this. This is our muscle, right? And what will happen if we shorten this muscle here, right? We're going to move that bone in that direction. But remember that we're also going to have a muscle back here, right? So if this is going to send a signal to the central nervous system, to the spinal cord, right? There is going to be an interneuron that connects this to an efferent muscle fiber that is going to come back, and it's going to inhibit the antagonistic muscle, right? Which is good, because if we're going to contract this or shorten this, we want to inhibit the antagonist, right? Because if we contract them both at the same time, that can lead to damage. So there's a reflex with the Golgi, or the muscle spindle, right, that leads to inhibition of the antagonistic muscle. Now, the Golgi tendon, right, this one's just a little bit easier, right, that we have sort of our tendon that's going to connect to our bone, and then we have our muscle. And I'm not going to separate this one into intrafusal or extrafusal, because for the sake of this argument, it doesn't matter, right? But the Golgi spindle organ sets down here at the junction of the muscle and the tendon, and that is going to send information to the central nervous system about the degree of stretch on that muscle and that tendon. So with these two, along with a variety of other sensory receptors that can detect tension in ligaments and in joints, which we collectively called proprioreceptors, or some people will just call them proprioceptors, right? Those are going to send information to the central nervous system about where you're at in space, so that it can help coordinate movements when you need to go and move.